Hello, everyone. My name is Stefan, and I'm a PhD student from the Australia National University. And I'm very grateful and happy to be presenting here today. And I'm talking about SWARM, which is a single molecule workflow for analyzing RNA modifications. And us bioinformaticians often analyze RNA-seq data. And when we do so, we often comprehend RNA through 4 different nucleotides, the A, C, G, and U, or as encoded in files as T. But that's ignoring the fact that there's more than 170 different RNA modifications, or in other words, there are more than 170 different letters in the RNA alphabet. And these RNA modifications together encompass the epitranscriptome. And the RNA modifications can be seen on ribosomal RNA, transfer RNA, and messenger RNA. They're deposited by the RNA-modifying proteins. And these modifications control many aspects of RNA, including RNA stability, structure, translation, processing, localization, and many, many more. The modifications can involve the writer, reader, or eraser enzymes. And these enzymes can have dynamic and context-based action, which is quite analogous to the epigenome. And it is no surprise that the modifications are involved in developmental processes, cancer, stress response, and many other biological and cellular processes, but also diseases. RNA modification truly came to public's attention last year when the Nobel Prize was awarded for the discoveries concerning nucleoside-based modifications that enable the development of effective mRNA vaccines against COVID-19, where particular chemical modifications of RNA have increased the translation stability, translation efficiency, and RNA stability, as well as reducing the immunogenic effects. And although epitranscriptome is extensively studied, we still have so many open questions regarding epitranscriptome. The primary question is which modifications are present in each mRNA transcript, or in other words, we're still struggling to detect RNA modifications. And from this struggle to detect them, there are so many other open questions, such as what is the modification dynamics across different species, tissues, environments, and also diseases? Then how is the RNA modification code maintained? What are the functions of the modifications? And what is the link to the tissue and cellular phenotypes? As I've hinted before, detection is still a very big challenge when it comes to RNA modifications. And the current conventional methods mainly rely on the chemical conversions or antibody and enzymatic reactions. And historically, these methods have had very low overlap in the detected sites between the different studies. And these methods remain very strenuous and quite resource intensive. And I think the biggest limitation is that these highly specialized protocols are restricted to one modification type per experiment. And this is a very significant limitation considering the overwhelming diversity of RNA modifications and also their dynamic nature. And fortunately, there is a very promising alternative for detecting RNA modifications, which is nanopore sequencing. And this technology is, to the best of my knowledge, the only commercially available technology which can capture native RNA with high throughput. And this is done by measuring ionic current as the native RNA is passing through the nanopore. And the measured RNA current, or as we refer it to as squiggles, can be traced back to the sequence and also RNA modifications. So in a nutshell, entire epitranscriptome is theoretically encoded within each direct RNA nanopore sample, which is quite puzzling. But the signals are quite complex and context-specific, so it is very hard to decipher the nanopore squiggles, and very sophisticated algorithms and machine learning approaches are needed to work with this sort of data. There have been numerous approaches for trying to detect RNA modifications from the nanopore. The simplest one rely on comparing a control sample with a direct sample, with the control sample usually being either a knockout of known enzymes or in vitro transcribed RNA, which has no modifications. And these approaches are still quite limited and quite resource intensive, and in my opinion, do not provide many benefits compared to conventional methods. So a more optimal approach would be to do simple single sample detection, which is much more resource efficient. And many of the nanopore-based modification methods are only limited to site-level detection, where they're not able to analyze the stoichiometry, or also known as the modification rate, at given sites, and also they impose difficulties for studying co-occurrence of different modifications. So the most optimal way to detect modifications from nanopore would be to do molecule-level detection at single-base resolution. And the forefront of such methods is CHUI, which can simultaneously detect M6A and M5C, and published in Eduardo's lab. And it's amazing you can detect 2 different modifications at the same time, but what about other 160 plus modifications? And here comes the project that I'm working on, which is a single molecule workflow for analyzing RNA modifications, where we start from the ground set by CHUI, and we expect for more modifications and also focus on improving the detection accuracy, bringing it to sufficient detection power for transcriptome-wide detection. And we've achieved that with major advancements in the model's architecture and also clever training strategies. And as of now, we can detect M6A, M5C, pseudouridine, and also AC4C from nanopore direct RNA sequencing. And before I dive into any quantitative benchmarks, I feel like this is the best visual way to explain what is Swarm actually doing and to illustrate the capacity of what we can do. So we are looking at an IGV snapshot, an integrated genome browser, where with Swarm, we can visualize multiple different RNA modifications in the same RNA molecules in the same transcript. And we're looking at the particular transcript with a sample from HeLa, and we're looking reads that are aligned to the reference. And in the same reads, we have modifications, particular modifications with different colors, with the red color being pseudouridine predictions and the blue color being the Swarm's M6A predictions. And you can easily see the standout red color on the left side, and they are stacked up on an individual loci, which is a pseudouridine site that's previously been detected by BitSeq, which is a conventional method for detecting pseudouridine. So our predictions coincide with the already known pseudouridine site. And then on the right side, we see the blue M6A predictions, which also stack up on top of each other, and they land at the known Glory M6A site, another conventionally detected M6A site. We do see some sporadic blue and red colors in the middle, but we can definitely see that the known sites very easily stand out from the rest of the background modification level. And some of the terms that I've been using so far, the site level detection would only give you the information whether a given site is modified at all or not. So you would not have anything above this reference track. And stoichiometry of the modification rate can easily be seen as the number of the detected molecules out of all tested molecules at a given site, essentially a modification rate. And how do we get there? How do we train models for detecting RNA modifications? Essentially, we're using a supervised learning approach where we start off from the nanopore signals with known modification states. And we do so with diverse nucleotide context, including both the different sequence contexts and also modification contexts. And as I've said, we start from the labeled data with the positives having the target modification or the negatives not having the target modification. And we are able to extract the nanopore, we are able to extract the features from the nanopore data. We vectorize it and we can pass it to a neural network. And in this case, we're using a residual neural network known as the ResNet. And after the classification layer, we are able to get the probabilities of the given signal having the target modification or not. And it's a number between 0 and 1, with numbers closer to 1, meaning that the model is very confident that the given signal has the target modification. And touching on the diverse training data that we're using, the backbone of our training data is in vitro transcribed RNA, where we can perfectly control the modification state of each molecule. And we have in vitro transcribed RNA with AC4C or M6A or pseudouridine or M5C or fully non-modified. But also in our training, we are incorporating some of the validated cell line data, including the validated ribosomal RNA sites, which is really a wealth of data for RNA modifications, where there's a highly dense and diverse set of modifications which have been extensively studied and validated. And also we are using some validated mRNA sites that have high stoichiometry and that also have been extensively validated. And with this, we can really show diverse contexts of RNA modifications and ensure that we have good generalization on our application cases. And we rigorously benchmark our tools. And all of the results that I'm showing are on independent samples that have not been seen during training, with not showing the sequence, nor the samples, nor the cell lines, if applicable, to the model. In this particular benchmark, we are looking at our results on the in vitro transcribed RNA, where, as I've said, we can perfectly control the modification states. And we are looking at the 4 different models, with the left one being the pseudouridine model, then the M6A model, AC4C, and M5C model. And each panel, we can see the predictions on different samples. So the green curve is on the left side is the pseudouridine model predicting on the pseudouridine sample, whereas the other colors, the red one being on the AC4C sample, the pink one on M5C sample, blue one on the M6A sample, and the gray one on the non-modified sample. And on the x-axis, as we are increasing the probability of prediction, which is the model's confidence that this is pseudouridine, meaning that 1 means that it's very confident. On the y-axis, we are looking at the positive prediction frequency, which is the proportion of the sample where we are predicting with the given threshold. We can see that as we increase this significance into this being pseudouridine, we are separating the green line more and more from the rest of the modifications. Essentially, we can really easily see that our model is really well separating pseudouridine from rest of the modifications, and when the probability is high, it is almost always predicting just pseudouridine. We can see the similar trend for M6A, where the blue line is very well separated from rest of the modifications, and the similar can be said for AC4C and M5C. So we can really see here that our models are modification specific, not just whether something is modified or not, but the specific modification type. And we do appreciate some differences in the modification specificity between different modifications, and we try to optimize this as much as we can. So I've shown some results on the in vitro transcribed data, but our models look really well on the experimentally verified mRNA as well, which is our sort of ultimate application. And here we are looking at the HeLa predictions for M6A, and we have extensively validated data set by GLORY, which is a conventional method for detecting M6A. And this GLORY method can also give us modification rate at each site. On the left panel, we are looking at the swarm's M6A rate compared to GLORY M6A rate, with each dot being 1 site. And we can see that the modification rate across, this was like a couple thousand sites, is really neatly correlating for swarm and GLORY. And just for comparison, on the right side, we have one of the state-of-the-art M6A detection tools, which is M6A base caller. And we can really see the difference between swarm and M6A base caller in terms of their alignment with the known modification rates. So I've spoken about the site-level detection, and swarm can achieve this by exploiting a distribution of modification probabilities to estimate the confidence of the site-level modification. And we can do this by training a neural network on predefined mixture of positive and negative IVT signals. And this is really useful for narrowing down the millions of sites in the transcriptome to only a few thousand or sometimes few hundred sites that are validated as being modified. In this benchmark here, we are looking at 4 different tools, Swarm, Chewy, M6ANet, and M6A base caller, where we've predicted with each tool on the HeLa transcriptome, and we've only selected the drag sites, which are the majority of sites that are known to be the motif that's modified by M6A. And as each of these tools gives us a way to threshold sites, we've selected thresholds to remain with 80% of sites that are validated by GLORY, and the 20% are not validated by GLORY. And when we fix this precision to 80%, we can see that Swarm is able to recover much more sites. Essentially, if you apply each of these tools on the same sample and you want to have the same stringency, same precision, Swarm will give you much more sites with the same level of confidence. And I think if you sum up these these sites by Chewy, M6ANet, and M6A base caller, you still get more sites from Swarm than from the other 3 combined. On the other side, if we look at pseudouridine, which is even more tricky for overlapping with the validated sites, for comparison, there's more than 100,000 known sites by GLORY, but for pseudouridine here, we only had 800 that are detected by 2 different methods, BitSeq and Praise. And here, to change up the task, we are looking at how many sites do we need to predict to recover at least 100 validated sites. So we are narrowing down the transcriptome as much as we can while keeping 100 validated sites. If you take tandem mod, you need nearly 200,000 predictions to have 100 validated, which is not very practical. If you take nanoSPI, you have 30,000, which is a bit better, but with Swarm, we can really narrow it down to 2,600. And we do note that we need still some room for improvement, but this is really bringing it possible to applying even models for low frequency modifications to the transcriptome-wide detection. And I've spoken so far only about the RNA-002 kit. Some of you might be using nanopore data, and there has recently been a change to RNA-004 kit. And we're still taking some time to generate all the core samples for really robustly training and testing models. But so far, we have trained some models with the data that we have available. And also with the new kit, Oxford Nanopore Technologies has started publishing their own models with Dorado, and we've benchmarked a Dorado version 0.5 with the M6A DRAC model with a comparable Swarm model for M6A. Well, our model was trained to do alt context, but we benchmarked it on an independent set of in vitro transcribed RNAs with M6A, and we've achieved higher detection power than Dorado with the single base, single molecule resolution. And if you look at this ROC curve, which is looking at the true positive rate over the false positive rate, the two Swarm models with different complexity of training data have achieved better area under the curve of above 0.99 compared to Dorado 0.98. But we note that Dorado still seems quite solid on this benchmark. And just to summarize the significance and why should you use our tool if you have nanopore direct RNA data, we are able to detect multiple RNA modifications simultaneously from single experiment with transcript level resolution. And we achieve unmatched modification specificity, which is robust for transcriptome level detection. And we have a framework which can explore RNA modification dynamics in countless contexts. These models can also be trained on any RNA modification, given that we have available data, and of course, they should be robustly tested to confirm what is the capacity of the models. And I think it's safe to say that Swarm enables a significant leap towards deciphering the dynamics and functional relevance of the epitranscriptome. I've put up here the QR code if anyone is interested to contact me to use our models before they're published. And I'll come back to that slide later. And to give the acknowledgements, many thanks to Eduardo for amazing support and guidance. And also many thanks to the collaborators, because this is not a one-man's work. This needs a team of people. So primarily thanks to Alias and Akanksha for the machine learning contributions. And also thanks to Nikolai and his lab for generating the data for us. Thanks for listening, and happy to get any questions.
Can you go by the microphone?
You mentioned it could be applied to other modifications as well. So if you have non-native modifications that you introduce by chemical agents, how much data would you really need to?
So it would depend on the application. If you have like if you modify every single nucleotide into that modification, like if you have a sample full of it, then you would need one sample where you have that modification introduced and one where you don't have it, like a positive and negative.
Yeah.
It should work quite well. But if you really want to have a transcriptome resolution where you have millions of sites that are not modified and only few that are modified, then you need really robust data to train and test it.